sequence. The leader sequence synthesizes or codes for leader mRNA. Leader mRNA, followed by, if it continues, then the whole mRNA from the structural gene, right? Now this leader mRNA has got 4 segments, 1, 2, 3, 4. Understood? The leader mRNA has got 4 segments, not leader DNA. Leader mRNA, the primary transcript product from the leader sequence, has got 4 regions. Now these 4 regions, they pair like this or this. There are 2 conditions. The first condition is when tryptophan is high. The second condition is when tryptophan is low. So when do we need tryptophan to be synthesized? When medium contains low tryptophan. We do not need to synthesize tryptophan when medium contains already tryptophan. So in the first condition, do we need to synthesize the structural gene? mRNA from structural gene? Yes or no? No. Means if we synthesize these, then this will synthesize enzymes which synthesize tryptophan. But tryptophan is high, so no need. We do not want these to be synthesized. So the leader mRNA will stop here. It will not go further. Fine. When tryptophan is low, the table turns. Then we need to continue the synthesis so that we can synthesize enzymes which will synthesize tryptophan. Clear? So leader sequence leads to formation of leader mRNA. Leader mRNA has got 4 regions, 2 conditions: high tryptophan, low tryptophan. In high tryptophan, we do not need to synthesize tryptophan synthesizing enzymes. In low tryptophan, we need to synthesize enzymes which are required for biosynthesis of tryptophan. Now come to the first condition. When tryptophan is high, we do not need to continue the transcription process. That means we need to terminate prematurely. That's why it is called attenuation. When it attenuates, when region 3 pairs with region 4, when region 3 pairs with region 4, see here, region 2, 3, 4. 3 has a part of sequence which is complementary to 2. 3 has a part of sequence which is complementary to 4. So whichever segment it will get free, it will then pair with that segment 3. Correct? So 3 and 4, if they pair, then transcription stops. Where 2 and 3, when they pair, then transcription continues. So which segment is important here? Out of 1, 2 and 3 and 4? Huh? 3 is important. 3 is important. Whether 3 gets 2 free or 4 free. If it gets 4 free, then it base pairs with 4. If it gets 2 free, then it base pairs with 2. Okay? So if 3 base pairs with 4, transcription stops. If 3 base pairs with 2, transcription continue. Correct? Now, transcription, the process of transcription in prokaryotes ends by 2 manner. One, formation of hairpin loop. Number 2, by row factor. Using row factor. Here, hairpin loop is utilized. Hairpin loop means the mRNA, the transcript, forms a hairpin within itself, which indicates the signal or signals the termination process of transcription. তার মানে hairpin loop form করলে transcription বন্ধ হয়ে যাবে. But this hairpin loop is very crucial. It should be formed in a proper manner. That should not be formed between 2 and 3. It should be formed between 3 and 4. Correct? Now we come, why here 3 base pair with 4 and why there 3 base pair with 2? Remember like this. It is very difficult to understand. If you remember like this, it is good. If you don't remember like this, then you have to mug up, or there are other ways to pass exams also. Okay, I am not the good one to teach that. Well, I am a better one to catch that. Fine. But if you don't get caught, don't think I am fool. Think I am benevolent. I'll let you do so. Because there's a dialogue of Salman Khan. Don't teach father. Don't teach father. Complete the sentence. Don't teach father. Huh? You don't watch movie? Hey, you don't watch movie. Don't teach father. কে পারবে? কি বল? Don't teach father how to make babies. बाप को मत सिखाना कैसे बाप बनते हैं, है ना? अब समझ में आया? मुंडी घुमा के बैठो. This is a tactics. Behave like I am very fool. Don't behave like that. You will become like that. है ना? Try to be smarter. What you are, show that. You are smart. I can make out. Don't try to be fool. Fine. Segment 4, end of segment 4, this region has got, remember, this is very difficult to understand. First thing you need to know, the transcription in prokaryotes, transcription and translation takes place simultaneously. Means before the whole transcription process ends, translation begins. That means what? DNA protein, DNA, transcription, mRNA, ribosome, peptide. So transcription and translation takes place simultaneously. So here, for synthesis of leader peptide, this is mRNA. The moment this leader mRNA is synthesized, it starts synthesizing slowly. Things are getting more and more complicated. Yeah? So we now have 3 leaders: leader sequence, leader mRNA, and the product, leader peptide. The leader peptide is a product of these all 4 segments of leader mRNA, starting from sequentially 1, followed by 2, followed by 3. So now when 1 and 2 is done, 1 forms with 2, no effect, base pair. 3 is formed. Now if tryptophan is high, what will happen? 4 will be synthesized rapidly. 4 will be synthesized rapidly. Why? Because end of this 4 sequence, this mRNA, at the end of the sequence 4, segment 4, there are multiple tryptophan residues, codons for multiple tryptophan residues. There are codons for multiple tryptophan residues. Now, if tryptophan is plentiful, then what will happen? Ribosome will not wait. It will go on synthesizing at its own pace. Fine? So, as soon as 3 is formed, segment 4 will be formed, completed, and 3 and 4 will base pair, and this will signal termination of the transcription, and leader mRNA will be separated and show the leader peptide. But in case of this condition where tryptophan is low, the tryptophan ribosome, what will happen? The ribosome will wait because tryptophan is low, so it will wait for tryptophan residues to come. It will go, then stop. Alright, tryptophan is not there. What will happen? Segment 4 synthesis will take longer time, বেশি সময় নেবে, কিন্তু by that time 3 is ready. 3 is ready. So 3 is free, segment 3 is free to base pair with 4 is still being synthesized slowly. It's tumbling. RBC is waiting when one tryptophan containing amino acid tRNA will come and it will synthesize leader peptide. But 4 is ready. 4 is not occupied by ribosome. So 3 will base, 4 is occupied by ribosome. It is not free. So 3 will base pair with 2. That means 3 now is unavailable to base pair with segment 4. Why? Due to delay in translation process. The product of translation is not required. What is required? The segment of segment 4 should be free from ribosome. But here ribosome is occupying the segment 4. Segment 4 is occupied by, it is synthesized, but it is occupied by ribosome. So it cannot base pair with segment 3. So what is free? Segment 2. So 3 base pair with segment 2. Now when segment 4 is synthesized, segment 4 lies alone, free. So this is not a signal for stopping transcription. So that will continue. That will continue synthesizing this mRNA, which will lead to synthesizing these enzymes, leading to biosynthesis of tryptophan. Correct? That's all. Is it simple? Not at all. It's very, very difficult, very complex to understand. But I told you, if you followed my words, I told you the key words to remember. If you follow the key words, it will be easy. I can guarantee you that nobody understood this thing in one class. Never. It is so difficult to understand because your brain should be very clear about the process, what is happening. Unless you are very clear about the transcription and translation processes, you cannot make out. Mere mugging up will not help to understand this particular mechanism. Probably that's why I am here to explain. It's not so easy. So here you have to remember 3 things. 1, the difference between DNA and RNA. 2, the leader mRNA, leader DNA sequence, leader mRNA, leader peptide. Leader peptide has nothing to do with the transcription termination process, but leader mRNA. Correct? 3 and 4 when the base pair segments on leader mRNA. When 3 and 4 base pair, transcription stops at high tryptophan. At low tryptophan, 3 is not base pairing with 4. Don't say 3 base pair with 2. That should be said like this. 3 base pair with 4, transcription terminates. 3 cannot base pair with 4, transcription continues. Don't say 3 base pair with 2. Transcription does not continue because 3 has base pair with 2. No. Transcription continues because 3 is unable to base pair with 4. Try to understand this difference. Fine. So that's all for prokaryotic gene regulations. There are 2 ways. One is attenuation, another is repression and de-repression. Same thing has been shown here. Now we come to quickly the eukaryotic gene regulation. You can take photographs also, I don't mind, because I have to finish by within 20 minutes. So there are 6 points. How eukaryotic genes are regulated. Prokaryotic genes, how many genes are there in prokaryotic? 100, 200, 300. In eukaryotic, few thousands. So very big genome. So regulation process is also very complicated. Correct? Eukaryotic genome is small, regulation process is simple. Eukaryotic genome is at every stage we can regulate. And we do not have those many stages in prokaryotes. Whereas eukaryote has got many stages. Correct? So these are the stages where it can be regulated. It can be regulated by at the stage of transcription, number 1. Number 2, borrow RNA processing, because prokaryotic RNA does not require processing. So if the processing is delayed or misled, then difference. Next, transport. Transport from where to where? Nucleus to cytosol. So each one has got an example. Nucleus to cytosol. The first one is very simple, transcription level. Second one, what is the example? RNA editing. Third one, what is the example? ALA synthesis by heme regulation. Fourth one, mRNA translation. Translation process can be regulated by translation inhibitors. Fifth one, mRNA degradation. What is mRNA degradation? The half-life of mRNA species. Some mRNA has got short half-life. So it will stay for a short period within our cell. So it will produce less amount of product. Some mRNAs are having long half-life. That means it will stay for long period in our cell. It will produce more products, more enzymes. Correct? Next, protein degradation. Every protein is synthesized, must undergo turnover. জন্মিলে মরিতে হবে, অমর কি কথা কবে। আমাদের মতন প্রোটিনের জন্ম আছে এবং মৃত্যু আছে। এই দুটোর মাঝখানে যে টাইমটা যদি ভ্যারি করে, তাহলে প্রোটিন ডিগ্রেডেশনের প্রসেসের উপর নির্ভর করবে। It will depend on the process by which protein is degraded. What are the different methods for protein degradation? Quickly. Phagosome. Next. By caspases. Next, by 26S proteasome. Right? Next, ubiquitin mediated pathway. Several ways of degradation of protein. Correct? So either of these process can be manipulated to lengthify or to prolong the half-life of the protein. Fine. So each one we will see with one example if possible. Here the different stages have been given. At this stage, second one is this stage, third one is this stage, fourth one is this stage, sixth one this stage, protein degradation. Same thing has been shown diagrammatically. You can take photograph. Correct? Done? Now, these are the ways by which genes can be regulated at the level of DNA, means transcription process. See? So many ways. What is that? DNA methylation, histone modification, gene deletion, gene duplication, DNA rearrangement, gene amplification, chemical activator, environmental activator. What do you call the environmental substances which influence our DNA? Epigenetic factors, which are not part of the cell. They are called epigenetic factors. Right? So these are chemical activators, environmental activators. These are epigenetic factors. Okay. DNA methylation. One example has been given. What is a cytosine? The cytosine, if it is methylated at this position, it becomes 5-methyl cytosine. Now, when it will be read by the RNA polymerase, it will be read as thymidine, not cytosine. So, if it was cytosine, actually it should have been given one guanine against it. But since it is altered and now it will be read as thymidine, so they will put one adenine. So guanine was there, now it is changed to adenine. Mutation takes place. Right? So methylation changes the transcription by introduction of mutation in mRNA. Does mutation in mRNA harmful? Is mutation in mRNA harmful? Answer is no. Why? Because mRNA does not carry over the genetic information from one generation to the next. It is a DNA whose mutation is dangerous. You know? Suddenly your son can become Virat Kohli. Just one mutation. You know? Then what will happen? কি করবি? কি করবি বল? বাংলা বোঝে না? হ্যাঁ? বোঝে শালা। তা তুই বাংলা বুঝিস, তুই কি করবি বল? হ্যাঁ? কোথায় পড়বি? শুটিং এ পাঠাবি? বাট ও তো বিরাট কোহলি হয়ে জন্মেছে। ছেলেকে বলে যা শুটিং কর, পয়সা নিয়ে আয়। ঠিক আছে, অনেকদিন হয়েছে ক্রিকেট খেলছিস, এবার শুটিং কর, পয়সা নিয়ে আয়। তাই না? আচ্ছা, এগুলি পড়তে হবে না, কষ্ট হবে, ছেড়ে দাও। Histone modification. How histones are modified? Histones are কি? Basic protein, তাই তো? কি করে এরা? Basic protein মানে they are positively charged and nucleic acid is negatively charged. So what they do? They cling to nucleic acid. So if you change the charge of histone proteins, then what will happen? They will not be able to hold on to DNA. So a new region will be opened where transcription will take place. Previously it was masked by the histone protein, now it is open for transcription. How you can change the charge of histone protein? By two ways: adding methylation, methyl group, that is neutral group, or adding negatively charged group, that is acetyl group. So histone now becomes neutral, histone now becomes negatively charged if it is acetylated. So now it cannot cling to nucleic acid. So the new region of DNA will be opened for transcription. Correct? Remodeling. See, এরকম ছিল, এরকম হয়ে গেল, এটা খুলে বেরিয়ে আসলো, এই জায়গাটা খুলে গেল, এই জায়গাটা আগে কি ছিল, এই জায়গাটা হিস্টোন প্রোটিন বন্ধ ছিল, এবার এখানে অ্যাসিটাইলেশন হয়ে গেল, হওয়ার পরে কি হলো, এটা খুলে গেল, এখানে আরএনএ পলিমারেজ বসলো, এই জায়গাটায় একখানা এমআরএনএ তৈরি হলো, যেটা আগে তৈরি হচ্ছিল না। কি কি এলিমেন্ট কাজ করে ট্রান্সক্রিপশন রেগুলেশনে? সিস অ্যাক্টিং এলিমেন্ট, ট্রান্সঅ্যাক্টিং ফ্যাক্টরস, domains of transacting factors. Domains means the regions where it binds with the nucleic acid. Domains. Hormonal regulation means addition of phosphate group, addition of acetyl group, addition of methyl group. This addition requires some enzymes. The enzyme should be synthesized by some gene, methyl transferase, acetyl transferase, phosphatase, phosphorylase. So these enzymes, if these are not synthesized, then who will attach methyl group, who will attach acetyl group to the histone protein? So these are also regulated by hormones. What are the examples of cis-acting elements? Promoters, terminators, enhancers, silencers, insulators. Right? PT-ESI. PT-ESI. These are the cis-acting elements. Remember. Can I pause details? This should be taught during transcription regulation. Transcription factors, RNA polymerase itself and sigma factor and other transcription factors. Do we have sigma factors? No, we do not have sigma factors. So here transcription factor means TF2D. TF2D. এগুলি দরকার আছে, লিখে রাখো। These are domains of the protein. There are certain domains through which the proteins can bind nucleic acid. Few of them has been mentioned here. What is that? HLH. Sorry, helix turn, helix. HLH, helix loop, helix. Zinc finger motif, leucine zipper, and homeodomain means same kind of structure. If it is cascade, if it is concave, it will be convex. Now these structural features should be present in the DNA or the protein. We are talking about the factors through which it will bind the nucleic acid. RNA processing, gene regulation by mRNA processing, exon shuffling, RNA editing, and mRNA longevity. mRNAs which contains shorter poly-A tail, there are few mRNAs which contain longer poly-A tail. It has been seen that the mRNA species with shorter poly-A tail have got short half-life. mRNA species with longer poly-A tail has got longer half-life. So by maneuvering the length of poly-A tail, we can shorten or lengthen the half-life of the desired mRNA species. Third point, বলেছিলাম allosynthese. ঠিক আছে? Example, transport of mRNA between compartments. Fourth point, mRNA translation. RNA interference, RNAi. Remember, these are the hot topics nowadays. You should be knowing more about this RNAi, miRNA, siRNA than the normal mRNA, because normal mRNA structural features and everything already been included in CBSE +2. So at this level, you should be more aware about miRNA and siRNA, and their use in medicine. Huge use they have. Okay. If we cannot cover this part, it's up to you. For SDL, you have to cover it yourself. Questions will come definitely in final. But usually we skip that because it contains 3 marks question, 4 marks question. Rest for the portion is more important. That's why. But there will be some question regarding this region. So it's up to you for self-study. Translation and control. So this is mRNA translation. One is mRNA RNA interference, translational control, and regulation by protein processing. mRNA degradation, and lastly protein degradation. Just go through. This is a pathway where it has been shown where protein is being degraded using ubiquitin mediated proteasome pathway. One of the pathway by which protein can be degraded. One of the pathway. Okay. That's all. Eukaryotic regulation is less asked, prokaryotic regulation is more asked, but eukaryotic regulation, certain parts are important. The examples, precisely saying that mRNA transport, give one example by which genes can be regulated by mRNA transport, maneuvering mRNA transport. So, example is allosynthase. Allosynthase is synthesized in the nucleus, correct, the mRNA, and unless it is transported back to cytosol, it cannot produce the enzyme allosynthase, and that requires heme. If heme is abundant in the cell, then heme will enter nucleus and it will block the passage of allosynthase mRNA. So, in absence of allosynthase mRNA, new allosynthase molecule will not be synthesized and heme synthesis will be inhibited. Right. This one example, good example to remember. That's all. Any questions so far? mRNA থেকে কি তৈরি হয়? প্রোটিন কোথা তৈরি হয়? সাইটোসলে। তাহলে mRNA যদি নিউক্লিয়াসে তৈরি হয়ে পড়ে, যদি সাইটোসলে ফিরে আসতে না পারে, তাহলে কি প্রোটিনটা তৈরি হবে? হবে না। এই পথটাকে বন্ধ করে দেয় কে? কিছু সাবস্টেন্স। এটা একটা পয়েন্ট অফ রেগুলেশন। এক্সাম্পল কি? অ্যালাসিনথেস। কে রেগুলেট করে? হিম। যদি মিডিয়ামে হিম বেশি থাকে, তাহলে হিম কি করবে? নিউক্লিয়াস থেকে সাইটোসলে এই যে mRNAটা আসছিল, ওটাকে বন্ধ করে দেবে। আসতে দেবে না। ঠিক আছে, যেবার মনে করে হিম কম আছে শরীরে, তাহলে হিম নেই, তাহলে অ্যালাসিনথেসের যে mRNA সেটা নিউক্লিয়াস থেকে বেরিয়ে সাইটোসলে চলে আসবে, সেখান থেকে অ্যালাসিনথেস তৈরি হবে প্রোটিনটা, এবং সে হিম সিনথেসিস করবে।